Hello everyone, here is Dr. Wasim for TBTOOL. Today I bring the tutorial about visualizing the gene structure using TBTOOL. What does gene structure mean? It means we want to check in a gene how exon and introns are distributed, which is called the gene structure, and what kind of motifs are present or which it code for, so that we can predict or know the function of that gene. Moreover, we can also analyze or visualize the promoter sequences present in the upstream region of coding gene, and also visualize the different types of motifs present within the coding sequences. So let's begin. So first of all, go to graphics. In graphics option, go to biosequence structure, Illustrator. Here you find a variety of options to visualize the gene structure. First of all, we will see the basic biosequence view. In this view, you need two files. The first one is a tab delimited format input file which code for gene or protein length. So here I already prepared a gene length file. So suppose the gene name is ABC and its length is 1000. So here I put three genes, C, D, E, its length is 1000, and E, G, F, length is 1000. Both these gene name and the gene length are separated through tab. So this is the basic format. Now upload this file here in gene and protein length tab, and the second option need set input ID list. So it is also has a format. First one is the gene ID, contain 4 columns. The first one is the gene ID, then start position, gene end position, and the domain. So here is the domain parameters I have put. So ABC contain a domain ARF which start from 253 and it ends to 300. Similarly, I put CDE and name it IAA and put its coordinates for the start or end position. Similarly, EFG, I named it as MYB and it starts from 120 and ends at 450. And then here are some other parameters like motif shape. If you want round rectangle, you can choose round rectangle. If you want just rectangle, choose rectangle. Fill gradient. So first we will check and then press start. So here you can see we have got our three genes A, B, C, C, D, E, E, F, G. We have these three genes, and the distribution of these domains within these genes is now visualized. So the color is in gradient. We can change the color, like we don't want gradient, we want full field. So now the color is changed and it's not showing in gradient, it's fully filled colors. The next, if we want to rename our genes, like we want to name ABC like ARF containing the ARF domain, so we name it ARF1, like CDE contain IAA, so we can name it IAA1. Similarly, EFG can be named as MYB because it contains MYB domain. So we need to input a rename file. So the rename file is also in a tab delimited format. The first column will contain the gene name, and the next second column will contain the renamed gene. Okay, like here ABC, I named it ARF1, CDE is named as IAA1, and EFG I named it as MYB1. So now let's load it to the rename column. So here in the rename tab, we load it and then press start. So now here you can see all the genes. ABC is renamed as ARF1, CDE is renamed as IA1, and EFG is renamed as MYB1. And you can rearrange the graph, like change these legends, the captions to the place you want. Like here I am putting on the top, so now they are on the top. Or you can also put in bottom, whatever the place you like, and then press save. Choose the format you need or you want to save or you wish to save, and then press save. Give it the path, and then it will be saved. So this one is the first option. In the next option is visualizing motif pattern. So this need a mask.xml or meme.xml file. How this file can be generated? Here we have the Tata website, memesort.org. In this database we can analyze, or in this tool we can generate this file. Just open this website and go to meme tool. After that this tab will appear. So here you do not need to change these two options. Go to the third option, input the primary sequences, and upload your protein sequences here. Like I have these protein sequences, so I upload it here. And then select the site distribution. How do you expect motif sites to be distributed in the sequence? So by default I choose 0 or 1 occurrence per sequence. And how many motifs you want to visualize, it depends on you. Normally I choose 10 or 15, so here we choose 10. You can also input your email address, job name, and then press start. After you press start, the window like this will appear, and you need to wait for some time until the job will finish. Here on the right side you can see in the recent jobs, all the jobs or the queries you have put will queue up here. So once it will finish, the results looks like this. It is in MEME, HTML format, XML format, text format, and in MAST, HTML, MAST, XML, and MAST text format. From here we need MEME XML. So when we click on this, we will get the tab like this. The tab will appear. So just right click and save as. Save it on the place you want. So I have already saved it here, MEME. So now we need to visualize. So just put it here and then press start. So here you can see our MEME result is visualized. So here is the gene name which contains the different motifs, and here are the motifs that are present in these genes. And this is the length of gene, and it will show like this. You can change the graph settings Like you can change the dimension here. I am putting 1200 by 1200 and press fresh, so you can see its dimensions are changed. So you can adjust the dimensions according to your requirements, and then press save graph, choose the image format you required, and then press save to save in the path or in the directory you wish. Okay. The next one is visualize domain pattern from NCBI, a batch search. So for this we need NCBI CDD table from NCBI CDD batch search and an input file in FASTA format. So how we can get NCBI CDD table? For this we need to go to NCBI website here. This website, NCBI batch web CD search tool. Here you can search on the conserved domain present within your protein sequences. Just simply choose your FASTA formatted protein sequences, and from adjust search options choose any database you wish to search, and then press submit. So it will start searching, and it will take some time. It depends on the number of queries, and then after it will finish, it will appear like this. So here are two options. The first one is you can browse the result, or you can download the results in a different formats. So here I am going to choose domain hit, data mode in concise, and then press download. After I press the download, I will get the file with name hit data, which contains the distribution or the coordinates or the position of domain which is present in our sequences. So we don't need to modify this file, just simply download this file and put it in tbtool set input NCBI CDD table file, option of visualize NCBI CDD domain pattern, and then the next you set input FASTA file. This is our protein file that we have used for visualizing or predicting the domain present in those genes, and then we can press start. So here we can see the domains present in our genes are visualized. I can change the dimension. Fresh. Close. So here you can see our genes contain grass domain or the grass super family domains. We can modify with a different option, like if we just want to see how many genes contain, or just want to see that only those genes that contain grass domain, then we need to input here. Here we need set retain domain list, so which we want, like we just want grass, then press start. So here this will show only the grass domain while mask other domains. So it depends on you how you want to visualize. In set rank seek ID list, you can also put here the sequence list you want to visualize. If you have more sequences, or the your hit data contain different domains, and just you want to visualize some particular domain, then you can set here the input gene ID list here, so that only those genes will appear in the next tab. Like I just want, suppose I want just these two, we don't want other, then I just put the gene ID of these two and then press start. So here you can see only those two genes appear here. Then we can save or use according to our new requirements. And the next one is visualizing gene structure from GTF or GFF. This one is basically used to visualize the exon and intron distribution within a gene. So for this we need a .gff3 or the GTF file of our genome and the gene list, or if we have the coordinates, start position, end position, or the domain we want to visualize, then we can put it here. It depends on us the way we want to use. So I just want to visualize the exon and intron present in my gene list, so I put here the gene list, and in the set input GTF file I will put my genome GTF file, and then I will press start. So here it will appear with a figure containing or this. So here you can see it's showing the UTR, if UTR is present, and the CDS. All these things are now showing here, and you can modify this graph, just change the dimension or whatever way you like, and then save it in any format you want. The next one is trace mask XML to motif. Like last time we have done MEME motif analysis, and here we got two types of files, MEME and XML and masked XML. We used MEME XML to visualize the motifs, but we don't know the sequences of these motifs. So we use masked XML output to check what kind of sequences or these motifs contain. So for this we can use this tool. We just need to input our mast.xml file and give it an output pathway for motif sequence information. So I named it motif seq.text, and also need an output pathway for the region information, that this motif present in from which to which place, and it will give that information. So this is the motif region information dot text, and then press start. So here it generated two files. One, the first file contains the domain, second its sequence, motif ID, its length, and some other values. So this one is the sequence represented by motif 1, and here this one is its length 50. Here is the motif 2, it's MEME2 and its length is 70. So same like this, all these motifs that we have searched, 10 motifs. So the 10 motifs along with their sequences are here. But we also want to know which motif is present in which gene. So the second file gives us this information. Like this, our first gene contain motif 7 and its location is from 92th amino acid to 98th amino acid. Similarly, this motif 8 is present from 422 to 458. Motif 3 is from 480 to 529. So all this information is given in this file, and then we can use it according to our requirements. The last one is very interesting. You can do all of your individual analysis together here in a single tool. So this need set NAVIC tree. You can put here your phylogenetic tree in NAVIC format, or you can even put your gene ID lists. You can also put your MEME results, GTF files. Here you can put your CDD results, biosequence results, and you can also rename. So I am going to show you how we can do this. So the first one here is set NAVIC tree. So I am putting here NAVIC tree. Second is MEME. So I am putting my MEME result. Third one is like it needs GFF3. With this GFF3 it will display the exon and introns. And here we can see in the append the CTD results. So this tab appears with CDD results. So here we need to input our hit data, and the below tab needs FASTA file, our protein sequences in FASTA format, and then press done. Biosequences, if we have any additional information, then we can put our information here. It needs sequence length. We just need to put this file containing two columns. The first column is the gene name and the second column is the gene length. And the domain information here, it will contain the information about the domain, which domain is present in which gene, and from where it starts to where it ends, and what is the name. So basically it contains the 4 columns. The first column is for gene ID, second is the domain start, third is the domain end, and fourth one is the domain name. And then press done. After that we can press start. So here you can see it displayed all the graphs together. The first one is the motif, second one is the domains present in these genes, and the third one is the exon and intron distribution. So I want to rename it, so I will import a rename file. This rename file is also tab delimited, contains 2 columns. The first column is for the gene IDs. These are the original gene IDs, and these are the renamed gene. So whatever the name you want to give, you can input here. And both of these columns are tab delimited. Remember it. And then press start. So here you can see all the genes are renamed. So we can adjust its size just 2 times. Firstly double click right and then press refer. You need to do it fast. Double press right and then left. So you can see here the red border will appear. Then you can adjust this border. Like I drag it here, then I am going to adjust all at the single level. So here you can see all my three graphs are now at the same level. Then press save, and it gives the different formats. Can save in any format according to the requirement. Okay, the last one is the promoter visualization. How we can visualize the promoter? First of all, we need plant care, this website. Plant care. This website is for cis-setting regulatory elements analysis. Just choose the option search for care. Here you can input your email address. You can rename, put your header or your query name, and here you can paste your nucleotide promoter sequences, and then press search. After some time, maybe it depends on the traffic on the server, they will send the result on your email. In the email you will get the results in zip format. Extract the zip and then find out .tab file. You can open, when you open this tab file, it's look like this. It contains 1, 2, 3, 4, 5, 6, 7 columns. The first column is for the gene ID, second is the what kind of promoter is present, its name, third is its sequence, fourth is for the position, and the fifth is for the length, and seventh is for whether it is present on sense strands or anti-sense strand, plus minus, and what kind of motif is this, or it belongs to which representative plant. Also here it looks like this. So you can choose according to your requirement or the interest you want to visualize. So I have just created an example file. So I am going to do it. So we need to go to basic file sequence view. Here we need set input gene position length, gene or protein length in tab delimited format. So these are the promoters. So the promoter I choose up to 1000. So I am putting like gene name 2 column. It contains 2 columns. The first column is for the gene name and the second column is for the promoter length. So I choose up to 1000. So I put here just 1000, and both columns are tab delimited. And this one is the set input ID list. This file also or should be in tab delimit, and it contains the 4 columns. The first one is the gene ID, second one is the start position of promoter, third one is the end position of promoter, and the fourth one is for the name of promoter. And here the other parameters, you can choose different parameters and the color how we want to see. Then press start. So it will visualize like this. See, I have mentioned just 2 genes. So the 2 genes appear here, showing the distribution of promoters in their promoter sequences in their promoter sequence. So here we can adjust all these legends according to our requirements. can see auxine, MBS, MEJ and ERE elements are present, at which position and in which gene contain which promoter. So you can adjust on the top or either at the bottom, depends, and then press save, choose the format you want. So this is for all today. If you haven't subscribed, please subscribe TBTOOL, and if you enjoyed the video, please give it a like. As always, thank you for watching. This is Wasim, and I will see you next time.